I'm George, but usually report these meetups in the region. And I recently ran into this article from publication of ACM about miraculous data structure named Suffix Trees. And there are people that dedicate a career studying this data structure. It looks like it has lots of applications. One of the applications is in the bioinformatics. I was thinking what example I could have of a theoretical endeavor that could have the biggest impact, or one of the biggest impact on society. The cancer endeavor is very big and it involves lots of professionals. One of them are computer scientists and computer science professionals. There are like biologists, statisticians, like big data specialists, for example, and many others. Yeah. So I was narrowing my research because this is like a work in progress. It's like kind of notes I made during this research. I was trying to show how a theoretical approach evolved into algorithms, and then how they've been applied in an effort to cure cancer. And let me start with a short introductory video. So this is a brand new program announced by NASA that has elements of bioinformatics in it.
I think we're all healthy most of the time, but there are times we get sick. Sometimes we get really, really sick, and the consequences can be quite serious. There are several problems with our health system. First, we actually treat people after they get sick. We actually don't do much to keep them healthy and understand what's going on while they're healthy. And the other is we're in a one-size-fits-all. We basically give people the same treatment, even though we're all individuals, and even though the way we may respond will be very different. So what I think we need to do is switch from this one-size-fits-all to something that's individualized, where every person is treated according to their genetics, all their life history, and their environmental exposures. There's an amazing revolution going on right now. Omics is really seeing the entire collection of molecules that make up you. Basically, it's your DNA, it's your proteins, it's all your metabolites and such. The sequencing of the human genome was a huge, huge major step in making all this happen. So for the first time in 2001, people were able to decode the DNA of a human being. And that basically set up a reference onto which we could actually do a lot of the mapping we're talking about. We can now sequence people's DNA and understand their genetic basis of disease at a level that's never been possible. Mass spectrometry lets us follow tens of thousands of molecules. And because of this, we're getting an IMAX-like movie of what's going on when people are healthy and when they're sick. In many respects, it's like a jigsaw puzzle. A jigsaw puzzle can be made of 1,000 pieces. You don't really see the picture until you put all those pieces together. That's the same for omics. You basically try and understand all the individual pieces so you can see the whole picture. NASA Twin Study is an amazingly unique study where we're studying Scott and Mark Kelly. In this study, we had one twin launching into space, undergoing incredible stress responses during launch and during the time he's living in space. And the other twin, the identical twin, is living on the ground a normal life. And really what's nice about omics is we're going to see a picture of both of these astronauts at a level that's never been looked at before. We're going to be making billions of measurements, the one on the ground and the other one in space. And we'll see what space really does to the human body. I think this study will be very valuable for understanding space travel because ultimately we hope to send people to Mars. And that's going to be a long period, probably several years. And by getting this IMAX type movie, what's going on is people see space radiation, space food, all the environmental impact of space. We'll be able to better treat, hopefully, astronauts and manage their health while they go on this incredible journey. Omics is really an amazing field where we can look at people and their health at a level that's never been possible before. And with that, we'll be able to better manage people's health and try and keep them healthy, again, long before they get sick.
From here, I just want to make a disclaimer that the suffix 3 don't cure the cancer. Yeah, they just are an element that's, I would say, very small. That's part of the whole effort. Yeah, and so let's start to see. So what are the suffix 3? This is a Wikipedia entry. It's a data structure on which algorithms have been built to solve, for example, at the beginning was solve to find a linear time solution to the longest common substrate problem. Here they give an example of suffix 3, and that contains actually all the suffixes. For example, for banana here, the terminal node is an index indicating the number of the suffix. So we have at the beginning the whole word. Index 0 is banana, and then we have starting from the tail up to B, we have anana, it's this one, index 1, and then index 2, 3. So this structure turned out it's very efficient to do quick searches in strings. And why we're talking about strings? Because the DNA is actually a 3 billion sequence of characters. So it's a string, a very big string. And when in bioinformatics, when we study the DNA and genetics and diseases and things like that, we have to do searches on the DNA string and do pattern matching. Here from Wikipedia, as I said, these are like research notes, so I'm just highlighting things that I found while researching. I found this article like a few weeks ago, and the last 2 weeks I'm just only obsessed with bioinformatics. So here it gives you a list of applicational suffix trees. They also, you see here, have application in data compression and other. For example, Burrow-Wheeler transform is also used in bioinformatics. And here we have an example of 3 individuals with their DNA, and we see that most characters are common, only there are a few differences that actually define the variation between individuals' features. When we study, for example, cancer, from what I read, a sample from the cancer tissue is taken, and its DNA is sequenced and then compared to a normal individual. But the challenges are quite big because there are lots of issues to see exactly what's specific about the cancer cell. And there are so many types of cancers and so many types of different comparison that needs to be done. I have another video where a Stanford course introduction I found quite useful to give a better picture.
Hi, I'm Mike Snyder. I'm looking forward to telling you about our New Frontiers in Cancer Genomics course. It's an exciting new area. There's lots of exciting things happening, and you're going to learn about them in this course. Cancer is a really, really important disease. 50% of men and 33% of women will get cancer at some point in their lifetime. We need better ways to diagnose cancer, and we also very sorely need better treatments. Cancer is a genetic disease. Almost all cancers are the results of multiple mutations. Cancer is also a genomic disease, in that every cancer cell has a unique collection of changes that cause cells to grow uncontrollably. If we really want to understand cancer and also treat cancer, we have to look very carefully at the DNA of the cancer. What we're going to learn in this class, the difference between inherited and sporadic cancers, so inherited cancers, the familiar cancers, the kind that run in families. We're also going to learn about classifying cancers based on the genome differences, not just where they come from. Very importantly, you're going to understand how cancer cells evolve to become resistant to treatments. There's a number of community initiatives for enhancing cancer research and understanding how cancer occurs. And finally, you're going to hear some very cool primary research that's occurring in the cancer field and some of the exciting developments that are going on. And certainly you're going to hear about current challenges in terms of trying to understand, treat, and research cancer. So overall, it's going to be an incredibly exciting course. There's so many new developments that you're going to learn about. Can't wait to get started.
I come back to the main article from the publication of ACM. With the whole story here, I'm coming back to theoretical stuff with the suffix trees. Donald Knuth, the legend of computer science, he had a conjecture to find the longest substring sequence that I mentioned before. Here I did a research about this problem along the substrings, how it is defined, how the suffix trees with a variation of generalized suffix trees solve the longest substring problem. Wiener came up with a linear algorithm, and he actually solved the Knuth conjecture with better performance because Knuth said that he conjectured the algorithm will be solved in n log n time, and Wiener came up with a linear algorithm. And later on, Craig here, he came up with a better solution to Wiener algorithm. If someone is not familiar, how they came up, both Wiener and Craig, through the miraculous serendipity, they came up with the solution to the algorithm from the theoretical point of view. Actually, Wiener came up with his algorithm like a byproduct to another problem he was working. And Craig also trying to understand Wiener's paper. I found this quite interesting. In the process of understanding the paper, he came up with his algorithm that was easier to understand, that was more elegant solution. Then from suffix trees, there is another data structure suffix array that has a better application in bioinformatics. And also here I found a reference to the theoretical side of suffix trace use. Here is the Barrow-Wheeler transform that has a great application in bioinformatics. To minimize the memory requirements, how Barrow-Wheeler transform is used. Here another application with overlaps and DNA analysis. The overlap issue is a challenge, and suffix trees come to the rescue. And as any theoretical endeavor, here I found an interesting quote from Karp, that's a well-known algorithmist that noticed how a theoretical approach was wrongly applied, found a flaw in the practical applications. I highlighted how it failed to deal with the repeats. I mean, the way it's interesting to enter in details. A nice picture about genetics and how the mutations come up, and how DNA variations. Here we have DNA from 4 individuals, and the variation in one individual is positive, while another one is negative.
Many children are born each year with a serious genetic defect, and in fact 25 million Americans will have some sort of genetic defect for a major disease that they'll encounter. There's been just a technological revolution. That is to say, we can now sequence people's DNA and understand their genetic basis of disease at a level that's never been possible. There's new equipment out there that lets us do this for the first time, and that's really just come about in the last decade. Before, this was an incredibly challenging task. It took 2,000 people about 10 years to be able to sequence the first human genome, and it cost somewhere between $0.5 to $1 billion. Now you can do it in less than 2 weeks for probably on the order of about $2,000. We all start out as a single cell, and we grow into a complicated human being. And your DNA is the instruction manual for making that happen. Your genome is your entire collection of DNA. And your DNA is really made of 4 chemicals or 4 letters that are strung together in a very particular sequence. So if we took all the DNA in our body and actually strung it end to end, it would take us all the way to Pluto and back. When you get your DNA sequence, you actually try and decode what the exact order of those letters are. And with that DNA sequence, you can then try and understand how different traits arise. Your genes are really the things that, in fact, make those traits like your eye color and your height. It also controls things like depression and autism. So your DNA really controls many aspects of who you are. What's amazing about the twin study is that we'll be able to tell what factors are causing environmental influence on a person and their health, and which ones are really genetic. They're identical twins, meaning they largely have the exact same DNA. And so when one goes into space, he's undergoing a certain environmental stress, undergoing the forces of space, whereas the other one's living a normal life on the ground. These guys are essentially genetically identical, so any differences they have are due to environmental encounters. If we're going to send someone on a 3-year mission to Mars, we really have to understand in detail what's going to happen to them and how to best keep them healthy. This will be incredibly relevant to us on Earth because essentially, as we see how people are living in space and the kinds of genetic factors that can help protect people in space, some of those same things will be very valuable for people on Earth. By reading your DNA, we hope to see what kinds of diseases you're at risk for, what kinds of genetic information protect you to certain kinds of stresses is really valuable just in general.